In this chapter, we are going to look at both the male and female reproductive system. First, we're going to start with some basic terms that are used to describe these structures. The word gonad refers to the organs that produce the gametes as well as the hormones. Ducts are designed to receive and transport the gametes. Accessory glands are any organ that secretes fluids into these ducts. Peritoneal structures collectively are known as the external genitalia. And the reproductive tract includes all of the chambers and passageways that connect ducts to the exterior of the body. Beginning with a brief comparison of the male and the female. Well, one thing we're going to notice is that they are functionally very different from each other. The female, for example, produces only one gamete per month. The female also is responsible for retaining and nurturing the developing zygote. The male, on the other hand, can produce large quantities of gametes throughout their lifetime. On average, a male can produce 500 million sperm per day. The main organ of the male reproductive system are the testes, otherwise known as the male gonads. The testes secrete the male sex hormones, referred to as androgens, and produce the male gametes, called spermatozoa or sperm. The gonads for the female are the ovaries. The ovaries release one immature gamete, also known as an oocyte, per month. The ovaries are also responsible for producing the female sex hormones. The female reproductive system also includes the uterine tubes. This is what carries the oocytes to the uterus. The uterus itself, which encloses and supports the developing embryo, and the vagina that connects the uterus to the external environment. We are going to start with the male reproductive system first, the various organs and their functions, and then we will transition to the female. First, we will start a little bit more in depth with the testes. The testes are an egg-shaped organ that hangs within the scrotum, and this is outside of the body. The scrotum is a fleshy pouch that is suspended inferior to the perineum, anterior to the anus, and posterior to the base of the penis. Now, the testes form inside of the body cavity during embryonic development, somewhere up around the area of the kidneys, and then they need to descend to the outside of the body to their permanent location within the scrotum. This process is helped along by a structure known as a gubernaculum testes. This is a bundle of connective tissue fibers that extends from the testes into pockets of the peritoneum. What this does is help lock the testes in position near the anterior abdominal wall as the fetus grows. Then, sometime during the 7th month of fetal development, this is a time when the fetus is growing very rapidly, as we will see in our final chapter, you will begin to see some circulating hormones, and these hormones will begin to stimulate contraction of the gubernaculum testes. What happens as the gubernaculum contracts? It pulls the testes lower and lower through the abdominal cavity, ultimately into the scrotum. So each testes will begin to move through the abdominal musculature, eventually outside of the body and into the pockets of the scrotum. As the testes is descending, it will carry various accessory structures along with it. These ultimately will form the body of the spermatic cord. These structures include the ductus deferens, testicular blood vessels, nerves, and lymphatic vessels. Let's look at the spermatic cord in a little bit more detail. Here on this side, we can see the spermatic cord, and on the other side, we see it opened up so we can visualize the structures inside. The spermatic cord extends between the abdominal pelvic cavity and the testes. It is made up of layers of fascia and muscle. It encloses various other structures, including the ductus deferens, blood vessels, nerves, and lymph vessels that supply the testes. It will pass through the inguinal canal and descend into the scrotum. The scrotum is where we find the testes. It is divided into two chambers, also called scrotal cavities, each of which will contain one testes. So each testes lies in a separate scrotal chamber, and it is covered by a structure known as the tunica vaginalis. Now, as we go through this chapter, we are going to see various vocabulary terms or structures that are labeled the same in both the male and the female. We will also, once we get to the female, see reference to certain structures in the female that are reproductively similar to structures we talk about in the male. Pay very close attention to all these different structures as we go through. The tunica vaginalis is one of these structures that we will also see used in the female when we get to the ovary. We can see it here. It's the outer covering of the testes. Another term, the raphe. This is a raised thickening on the scrotal surface that marks the partition of the two scrotal chambers. So it's the indentation here, and we see like a septa between that separates the two chambers. Another structure associated with the testes is the tunica albuginea. This too is a term we will see when we talk about the ovaries in the female. The tunica albuginea is deep to the tunica vaginalis. It sends fibers of itself that extend into the testes and will form fibrous partitions, also known as septa. These septa all then converge near the entrance to the epididymis, and we'll see these on our next slide, the next picture. Also around the outside, associated with the scrotum, is a muscle called the dartos muscle. This is a layer of smooth muscle that is found in the dermis of the scrotum. The dartos muscle is what causes the characteristic wrinkled appearance of the scrotal surface. All right, now we're going to look inside. So here, let's identify the structures first. We can see the tunica vaginalis forms two layers here around the outside, and then just inside, deep to the tunica vaginalis, is the tunica albuginea. The tunica albuginea sends extensions of itself through the testes. These are known as septa, and the septa here converge in this structure. So the septa are responsible for subdividing the testes into smaller units known as lobules. But each lobule can contain upwards of 800 slender, tightly coiled tubes known as the seminiferous tubules. The seminiferous tubules is where the sperm are actually produced, and each of which will form a loop that's connected to our next structure up here at the top, known as the rete testes. The testes also has between 15 and 20 efferent ductules, efferent exiting ductules. We see them labeled up here at the top of the rete testes. These efferent ductules connect the rete testes to the epididymis. Now here we're looking at a cross-section through one of the seminiferous tubules. And what we see around the seminiferous tubules, we will find a connective tissue capsule. And then around or associated with this capsule, we will find blood vessels. And then along the outside, we find some special cells known as interstitial cells, also called cells of Leydig. These are the cells responsible for producing the androgens or the male sex hormones, of which testosterone is the most important. We said the seminiferous tubules is where sperm are produced, so we are going to talk about this process. The term spermatogenesis refers to the process of sperm production. Sperm production begins in the outermost cell layer of the seminiferous tubules and proceeds toward the lumen. So the very beginning stages would be here around the outside, and as they mature, they get closer and closer to the lumen, or the center portion of the seminiferous tubule. There are 5 basic cells involved in the process of spermatogenesis, and again, they start The most undifferentiated ones are out here, and the closer they get to the lumen, okay, the more mature or closer they're going to look to the actual sperm. Also look in this picture. This is a seminiferous tubule. Here's some of the connective tissue capsule. This out here is where we find the interstitial cells, or the cells of Leydig, as well as some blood vessels. The first cell type are called spermatogonia. These are the stem cells that are going to give rise to the sperm. They divide by mitosis to produce two daughter cells. And these cells retain the ability to constantly produce, or divide by mitosis, to continually produce new cells throughout the life cycle of a male. So the spermatogonia undergo mitosis, producing two daughter cells. One of those daughter cells remains at the level of the spermatogonia, so it can continue to divide. The other one moves up a level and becomes a primary spermatocyte. It is the primary spermatocyte, this next layer, where meiosis begins. When they divide, go through the first division of meiosis, they will form a secondary spermatocyte. The secondary spermatocyte ultimately differentiates into spermatids. A spermatid is an immature gamete. Spermatid then differentiate into spermatozoa. Spermatozoa, over time, as they finish their maturation process, will ultimately lose contact with the wall of the tubule and enter into the fluid of the lumen. This is showing the process here. Here's our spermatogonia that undergo mitosis, and they will produce two daughter cells. One stays behind, the other moves on to become a primary spermatocyte. This primary spermatocyte enters into meiosis, forming a secondary spermatocyte, ultimately two spermatids. These spermatids technically are sperm, but notice they do not look like sperm. They need to finish the process, and this is known as spermiogenesis. Spermiogenesis is the last step of spermatogenesis. Each spermatid needs to mature into one spermatozoon, or sperm. As this maturation process occurs, the spermatid will not be free. They will remain attached and surrounded by the cytoplasm of a special type of cell known as a nurse cell. Nurse cells do exactly what their name implies. They support and nurture these maturing sperm as they undergo this process. So again, the functions of these nurse cells support spermiogenesis, this final development process. They also secrete a hormone known as inhibin. This hormone, inhibin, inhibits the production of FSH and gonadotropin-releasing hormone. This gives feedback control over the spermatogenesis process. We will look at these hormones in more detail in just a moment. The nurse cells also secrete androgens, androgen binding protein, which helps to stimulate spermatogenesis. Androgen binding protein, this is where the testosterone, which is the androgen, would bind to this protein to stimulate the process of spermatogenesis. Let's look at the structure of a sperm once they are finished. Each sperm will have a head. This is where you find the nucleus. At the tip of the head, there is an acrosomal cap. This cap contains enzymes that are essential to the fertilization process. Just behind the head, there will be a neck. This attaches the head to the middle piece. In the middle piece, you find many mitochondria. Remember the function of mitochondria. They produce ATP. This is the area that will produce the energy that the sperm is going to need to help support its movement. And then finally, there is a tail or a flagellum that once they are finished their total maturation process will allow them to swim or to move. Spermatozoa, once they are mature, will detach themselves from the nurse cell. At this time, they will be free in the lumen of the seminiferous tubule. However, they may look mature, look like they are finished, but they are functionally immature. Structurally, yes, they are finished, but functionally, they are immature. They are incapable of locomotion or fertilization. In other words, they cannot move on their own, and at this point they would not be able to fertilize the female egg. They are moved along by cilia that line the efferent ductules into the epididymis. They cannot move on their own, so they have to be pushed along using the cilia. Our next structure is the epididymis. This is the start of the male reproductive tract, or the various tracts that are going to move the sperm. This is a coiled tube roughly 7 meters in length that is bound to the posterior border of the testes. So we see it here coming around the posterior portion of the testes. Functions of the epididymis: it will monitor and adjust fluids that are produced by the seminiferous tubule. It also monitors the sperm and will recycle damaged spermatozoa. It will also store and protect the spermatozoa to facilitate their functional maturation process. When spermatozoa leave the epidermis, they are mature, but they are still immobile. To become motile, in other words, in order to actively swim, they must undergo a process called capacitation. Capacitation involves 2 steps. First, the spermatozoa become motile when they are mixed with secretions from the seminal glands, which we'll talk about here in just a moment. The spermatozoa become capable of fertilization when they are exposed to the female reproductive tract. Then different changes happen, and they are then capable of fertilizing the egg. From the epididymis, we go into the ductus deferens, also known as the vas deferens. This is a 40 to 45 centimeter long tube that begins at the tail of the epididymis and then ascends through the inguinal canal towards the bladder. It then comes behind the bladder toward the prostate gland and the seminal gland. So we see it right here as it's coming around and down behind the bladder. As it approaches the prostate gland, it enlarges, and this is known as the ampulla near the end of the ductus deferens. The ductus deferens is lined with ciliated epithelial tissue, and it will undergo peristaltic contractions. Remember, the sperm cannot swim on their own. They need to be pushed along. So the combination of the cilia and the peristalsis will move the sperm through this tube. The vas deferens is also capable of storing the sperm upwards of several months prior to ejaculation. What we see here at the end of the vas deferens, it is known as the ejaculatory duct. This is a short passageway at the junction of the ampule and the duct coming out of the seminal gland. The two join, and this forms the ejaculatory duct. The ejaculatory duct penetrates the wall of the prostate gland and ultimately empties into the urethra. The urethra makes the last portion of the male reproductive tract. The male urethra is used by both the urinary and the reproductive system. It extends from the urinary bladder down to the tip of the penis, and it is divided into 3 regions. The prostatic portion is the part that goes through the prostate gland. The membranous is the middle region, and then the spongy, also called the pineal urethra, is the portion through the penis. Look here at the prostate gland. We can see here's our seminal gland, and it's hard to see where the vas deferens come in, but the two join in this structure right here coming out of the seminal gland. That is the ejaculatory duct that comes down and joins with the prostatic urethra. Next, we are going to look at various accessory glands associated with the male reproductive tract. The functions of these glands is to activate the spermatozoa. They also provide nutrients for the spermatozoa that they're going to need ultimately for motility. Also, the sperm can be stored for many months, and they have to be fed during this time. These accessory glands also help to propel the spermatozoa and fluids along the reproductive tract through peristaltic contractions. And they also produce buffers. This helps to counteract the acidity of the urethra as well as the vaginal environment. The first of these accessory structures are the seminal glands, or also called the seminal vesicles. These are tubular glands, and they're coiled and folded into roughly a 5 centimeter by 2.5 centimeter mass. These are very active secretory glands responsible for producing roughly 60% of the total semen volume. The fluid that is produced by the seminal glands, its composition, it contains a very high concentration of fructose. This is a sugar. What this does is feed the sperm. It also contains prostaglandins. Prostaglandins will stimulate contractions of smooth muscles in both the male and the female reproductive tract. In the male, this will help with peristaltic contractions to move the sperm, and in the female, it will cause contractions in the uterus to help guide the sperm in the correct direction. It also contains fibrinogen. Fibrinogen is responsible for forming a temporary clot in the female reproductive tract. The secretions from the seminal gland are slightly alkaline. This helps to neutralize the acids in the prostate gland as well as in the vagina. These fluids initiate the first step in capacitation. Once the sperm are exposed to these fluids, they will begin beating their flagella and will become highly motile. The secretions from the seminal gland will be discharged into the ejaculatory duct at the time of emission, and this would be when peristaltic contractions are underway. Next structure is the prostate gland itself. This is a small muscular organ approximately 4 centimeters in diameter. It encircles the proximal portion of the urethra just below the urinary bladder. So again, you can see how it surrounds the urethra, and this is why males, when they have prostate problems or the prostate becomes swollen, it will compress the first portion of the urethra. This will prevent the bladder from being able to empty itself. The fluid from the prostate gland, this is responsible for producing another portion of semen, is slightly acidic. It comprises between 20 and 30% of semen volume. Just like the fluid from the seminal glands, it will only be ejected into the prostatic urethra by peristalsis at the time of ejaculation. Another accessory gland are the bulbal urethra glands. They were once upon a time referred to as the Cowper glands. You can see them here on either side. These are compound tubular mucus glands. They are round-shaped glands right at the base of the penis. They secrete a thick alkaline mucus. This helps to neutralize the urinary acids in the urethra, as well as helping to lubricate the glands, which is the tip of the penis. This will empty into the lumen of the urethra also at the time of ejaculation. All these glands are responsible for producing what is known as semen. A typical ejaculation will release between 2 and 5 milliliters of semen. This includes the sperm, the seminal fluid, and various enzymes. Abnormally low volume of semen may indicate problems with either the prostate gland or the seminal gland. Another thing that can be done is a sperm count. If couples are having problems with fertility, they would do a sperm count to see if the male is producing the normal volume of semen as well as the normal number of sperm. A sperm count will be taken of semen collected after 36 hours of sexual abstinence. Normal ranges for sperm should be between 20 million and 100 million sperm per milliliter of semen. Next, we will look at the male external genitalia, which includes the penis. The penis is a tubular organ through which the distal portion of the urethra will pass. It is designed to conduct both urine and semen to the exterior and introduce the semen into the female vagina. Parts of the penis: The root is the upper portion, also known as the crus. This is the fixed portion that attaches the penis to the body wall. The body itself is known as the shaft. It contains 3 cylindrical columns of erectile tissue, which we can see here in cross-section. 2 are known as the corpora cavernosa, and 1 is the corpus spongiosum. At the tip of the penis is the glans. This is the expanded distal end that surrounds the external urethral orifice. The prepuce, also known as foreskin, this is folds of skin that surround the tip of the penis. This is what is typically removed during circumcision. Next, we're going to look at these columns of tissue. First are the corpora cavernosa. These are 2 cylindrical masses of erectile tissue that are separated by a thin septum. They diverge at their bases, as we can see here as they come up. They diverge at their base, forming the crux of the penis. They are erectile tissue, and each of them, the tissue surrounds a central artery. The third column is the corpus spongiosum. This is a slender erectile body that surrounds the pineal urethra. It extends from the urogenital diaphragm down to the tip of the penis. And then this is what is responsible for expanding to form the glands. We're going to look at the hormones of the male reproductive system. Everything begins with gonadotropin-releasing hormone. This is synthesized by the hypothalamus, released from the hypothalamus, and travels to the pituitary. This controls the rate of secretion of both FSH and LH. The other male hormone is testosterone. Testosterone typically is released in response to LH. FSH and testosterone both target the nerve cells of the seminiferous tubules to promote the process of spermatogenesis. Hormone secretion is regulated by the process of negative feedback. We looked at this back in the endocrine chapter. Spermatogenesis is regulated by 3 major hormones: gonadotropin-releasing hormone from the hypothalamus, FSH from the pituitary, and then inhibin. So so we can see here, FSH, okay, comes to the seminiferous tubules, targets the nerve cells, and would cause them to secrete inhibin. So as the process of spermatogenesis accelerates, inhibin secretion will increase. And then what inhibin does comes around, negative feedback. It will inhibit FSH as well as suppress the secretion of gonadotropin-releasing hormone. Inhibin and FSH together, elevated levels of FSH will end up increasing the production of inhibin, and inhibin levels will increase until the FSH levels return to normal. If FSH declines, so too will inhibin production fall, and this will allow FSH production to increase. The other main hormone from the anterior pituitary is luteinizing hormone. This one targets the interstitial cells of the testes and will promote the secretion of testosterone as well as other androgens. Testosterone then will target the nerve cells to promote spermatogenesis. Now, testosterone is the most important of the male androgens, or the male sex hormones. This is the main hormone responsible for stimulating spermatogenesis. It also affects the central nervous system. It has an effect on the libido or sex drive and other related behaviors. It stimulates metabolism in the male, and it helps to establish the male secondary sexual characteristics. This includes the distribution of facial hair, an increase in muscle mass and body size, and the characteristic adipose tissue deposits associated with the male physique. Testosterone also is responsible for maintaining the accessory glands and organs of the male reproductive tract. Testosterone is also important during development. Production of testosterone begins around the 7th week of fetal development and will reach its peak after about 6 months. This early surge in testosterone levels will stimulate the differentiation of the male duct system and the accessory organs, and will also have an effect on the development of the central nervous system. Testosterone helps to program the hypothalamic centers that will control gonadotropin-releasing hormone, FSH, and LH secretion, sexual behaviors, and sexual drive. This testosterone during fetal development, too, is also what is responsible for stimulating the descent of the testes. Next, we're going to look at the female reproductive system. Female reproductive system produces the sex hormones and functional gametes just like the male does, but the female has a few additional functions. One is to protect and support the developing embryo, and then to nourish that newborn infant. The female reproductive organs have a lot of structural support. The ovaries, uterine tubes, and uterus are all enclosed in a large ligament known as the broad ligament, and the uterine tubes here run along the top portion of that broad ligament. We're going to start with the ovaries, which are the female gonads. They are small, almond-shaped organs near the lateral walls of the pelvic cavity, and they have three main functions. One is the production of the immature female gametes, also known as the oocytes. The ovaries are responsible for the secretion of the female sex hormones, which include estrogen and progesterone. Just like in the male, they secrete a hormone known as inhibin. This is involved in the negative feedback control of FSH. Looking at the structure of the ovary, the visceral peritoneum is also referred to as the germinal epithelium. This covers the surface of the ovary, and right underneath it is the tunica albuginea. Tunica albuginea, same term that we saw that was referred to the outer covering of the testes. The stroma refers to the interior tissues of the ovary, and it is divided into a cortex region around the outside and a medulla in the center where you find various blood vessels. The gametes are produced here in the cortex region. The ovarian cycle includes monthly oogenesis that occurs between puberty and menopause. The ovaries have roughly 2 million primordial follicles at the time of birth, each of which contains one potential primary oocyte. However, by puberty, when oogenesis begins, this number drops to roughly 400,000. So from 2 million down to 400,000. Let's look at the process of oogenesis. The primary oocytes will remain in suspended development until puberty. At puberty, what stimulates this process will be rising FSH levels. This will trigger the start of the ovarian cycle. And then each month thereafter, roughly 10 primary oocytes will be stimulated to begin this process, but usually only one ends up completing the process. Two basic characteristics of meiosis in oogenesis that are particularly different, especially from what happens in meiosis in the male. The cytoplasm of the primary oocyte divides unevenly. It produces one ovum with the original cytoplasm and 2 or 3 polar bodies that ultimately disintegrate. Going back to this picture, okay, we have one primary oocyte that goes through the process. It will produce one ovum and anywhere from 2 to 3 of these polar bodies. The second big difference is that the ovary releases a secondary oocyte, not a mature ovum. The secondary oocyte will be suspended in metaphase of the second meiosis division. Meiosis will not be completed until the egg is fertilized. If the egg is not fertilized, it will never become a mature ovum. It will always stay in metaphase of meiosis II. Looking at the ovarian follicles, these are specialized structures in the cortex of the ovaries. This is where the oocyte grows and meiosis I will occur. These primary oocytes are located in the outer portion of the ovarian cortex, near the tunica albuginea, and they are found in clusters that are called egg nests. So we can see these clusters of primary oocytes. It's a little bit better picture here. We can see a nest egg right here. These primordial follicles, each primary oocyte in an egg nest is surrounded by follicle cells. So here's a primary oocyte, and you can see a thin layer of follicle cells around it. This primary oocyte and the follicle cells around it, this is what we refer to as a primordial follicle. Now, these are then going to be stimulated to continue through the ovarian cycle. So in response to FSH, what will happen is we will get the formation of a primary follicle. What happens? The transition, the follicular cells are going to enlarge and divide, forming several layers. There will be a layer known as the granulosa cells. These are follicular cells that have become more rounded, and these are also the ones that are closest to the oocyte. The other layer are the thecal cells. These are further out. They are formed from the granulosa cells as they enlarge and multiply. Both of these cell types can produce and do produce estrogen. You will also see the development of a structure called the zona pellucida. This is a glycoprotein-rich layer that we'll talk more about in our last chapter when we talk about the fertilization. I also want you to pay attention to the magnification. This is our primary follicle. Look at how much we're magnifying it. Back here, look at the magnification, how small these primordial follicles were, how much we had to magnify them by, versus here. They're much larger, and yet our magnification is less. We then transition from a primary follicle into a secondary follicle. Here, the follicular wall will begin to thicken. The deeper follicular cells will begin secreting follicular fluid that begins to accumulate in pockets, and we can see that through here. Eventually, this separates an inner and outer layer of the follicle. From the secondary, we transition into a tertiary follicle. By now, the oocyte is projecting into a structure known as the antrum. The antrum is a fluid-filled cavity surrounded by granulosa cells. They form a secondary oocyte by this stage that, remember, stays in metaphase of meiosis II. Also, look at the magnification now, only 136 times. Here's our secondary oocyte. Again, pretty significant in size. The oocyte and a layer of granulosa cells begin to drift free of the antrum, and it will begin at this stage to look almost like a pimple on the surface of the ovary. These granulosa cells that stay associated with the oocyte are referred to as the corona radiata. Now, when a woman who's trying to become pregnant and undergoes fertility treatments to have her eggs harvested for in vitro fertilization, how do they know where these eggs are? You can see them. The mature or the developing follicles, like I said, will appear almost like pimples on the surface of the ovary. So they can go in and they can harvest. They know exactly where these oocytes are. And then finally, this leads us up to the process of ovulation. This is when the tertiary follicle releases the secondary oocyte. The follicular wall will rupture, releasing our secondary oocyte and that corona radiata. This happens due to a surge in luteinizing hormone that happens right around day 14 of the female cycle. And we will look at this in more detail in a moment as well. All of this surrounding the egg that we see out here, that is the follicular fluid that was contained in that antrum. After ovulation, this is what that follicle looks like. We get the formation of what's called the corpus luteum. The empty tertiary follicle now collapses on itself. The granulosa cells will invade and fill up that anther. And this structure, the corpus luteum, will begin to produce another hormone known as progesterone. The purpose of progesterone is to begin to prepare the uterus for possible implantation. Now, if fertilization and implantation does not occur, the corpus luteum will transition into the corpus albicans. This represents scar tissue on the ovary, and the corpus albicans no longer retains the ability to produce hormones. Moving on to our next structure, the uterine tubes. They are also called the fallopian tubes or the oviducts. These are a hollow muscular tube that average about 13 centimeters in length. These tubes are responsible for transporting the oocyte from the ovary to the uterus. The various parts. First is the infundibulum. Now it's tough to see here in this picture. The infundibulum is like this expanded funnel near the ovary with fimbriae or like finger-like projections. What it does is come down and surrounds or kind of covers the ovary almost like an umbrella. The infundibulum does not touch the ovary. Some of the fimbriae may, but the infundibulum does not touch the ovary. There is a gap between the ovary and the opening of the uterine tubes. This is why when you look at that picture that we saw of ovulation, it looks like the egg is really being shot off the surface of the ovary. And it actually is, because it's got quite a bit of distance to have to travel. And it's not 100% given that when the egg is released, it will make it into the opening of the uterine tubes. Now, the inner surface of the infundibulum is lined with cilia, and these will beat with a rhythmic pattern that creates these currents that help to draw the egg toward the infundibulum, and then once in, will help to move the egg toward the middle segment of the uterine tube. The ampulla is this middle portion, and then the isthmus is the shorter segment between the ampulla and the uterine wall. It's the isthmus that opens into the uterus. The transport of the oocyte involves the ciliary movements as well as peristaltic contractions in the walls of the uterine tubes. A few hours before ovulation, the cilia will literally be turned on in this beating pattern, and peristaltic contractions will be initiated. Like I said, this will create currents that will direct and guide the egg that was just released into the opening of the uterine tubes. From the infundibulum to the uterine cavity normally takes roughly 3 to 4 days for that egg to make its way down. Fertilization, if it occurs, for fertilization to occur, the secondary oocyte must meet the spermatozoa during the first 12 to 24 hours after ovulation. Since this journey down the uterine tubes takes between 3 and 4 days, fertilization typically occurs near the boundary between the ampulla and the isthmus. So the egg fertilization occurs in the uterine tubes, and then it takes almost a week from that time to implantation. And again, this is all things that we'll talk about in our last chapter. Next structure is the uterus. This is a pear-shaped organ, roughly 7.5 centimeters long, 5 centimeters in diameter, weighing between 30 and 40 grams. Normally, the uterus bends anteriorly near its base. This is called anteflexion. However, in some women, it undergoes retroflexion. This is when the uterus is tipped backwards. Parts of the uterus, there are 3 main suspensory ligaments that hold the uterus in place. We only see 2, the broad ligament and the round ligament here in this picture. The uterine body is the largest portion, and it ends at the isthmus where the uterine tubes enter. And then this upper rounded portion of the uterus is known as the fundus. This is the superior attachment of this is superior to the attachment of the uterine tubes. The cervix is the inferior portion of the uterus. It runs from the isthmus of the uterus, okay, not the isthmus up here, but the isthmus of the uterus, to the vagina. The distal end of the cervix projects about 1¼ centimeters into the vagina. The uterine wall has a very, very thick layer, muscular layer known as the myometrium. And then it has a thinner, more glandular layer known as the endometrium. And then surrounding the whole structure is the perimetrium. So 3 layers. Inner layer is the endometrium, the thick muscular myometrium, and then the connective tissue outer wall known as the perimetrium. Estrogen is the hormone that causes the uterine glands, blood vessels, and epithelium to change with the various phases of the monthly uterine cycle. The myometrium is the thickest portion of the uterine wall and accounts for about 90% of the mass of the uterus. The function of the myometrium is to provide the force that is necessary to move the fetus out of the uterus into the vagina during labor and delivery. The inner layer, as I said, is the endometrium. This has 2 functional portions to it. The outermost layer is known as the functional zone. This is the layer closest to the uterine cavity, and this is the main portion of the endometrium that undergoes changes in thickness and structure during the menstrual cycle. Just behind that is a thinner portion of the endometrium known as the basilar zone. This is like the connection area between the functional zone and the myometrium. Now, the endometrium undergoes cyclical changes through the monthly cycle. The basilar zone remains relatively constant throughout the cycle. It's the functional zone that undergoes the most changes. And it undergoes these changes in response to levels of the various sex hormones, and it will produce the characteristic features of the uterine cycle. So let's look at this uterine cycle, also known as the menstrual cycle. This is a repeating series of changes that occur in the endometrium, and there are 3 main phases: menses, the proliferative phase, and the secretory phase. The menstrual cycle lasts between 21 and 35 days, depending on the female. Looking at these phases in more detail, first being menses. This represents the degeneration of the functional zone. This is caused by constriction of the arteries that will reduce the blood flow, oxygen, and nutrients to this layer of tissue. What happens is that the arterial walls will become weak, and they too will also rupture. This is one main source of the blood that is seen. This blood will be released into the connective tissue of the functional zone. Because these tissues are no longer receiving oxygen and glucose, they begin to degenerate and start to break away from the entire uterine lumen. During menses, the entire functional zone will be lost. Menstruation itself is the process of endometrial sloughing. This will last between 1 and 7 days, again depending on the female. Total volume that is shed will be between 35 and 50 milliliters of blood. The next phase is the proliferative phase. Now the epithelial cells of uterine glands will begin to multiply and spread across the endometrial surface. What this is doing is attempting to restore the integrity of the uterine epithelium. This will completely restore the functional zone during this phase. The proliferative phase is stimulated and sustained by estrogen that is being secreted by the developing ovarian follicles. And then the last phase is the secretory phase. Here, the endometrial glands will begin to enlarge and increase the rate of secretion. The secretory phase begins at ovulation and will continue as long as the corpus luteum remains intact. The secretory phase peaks about 12 days after ovulation, and then if fertilization does not occur, glandular activity will begin to decline. The secretory phase generally lasts for 14 days. It ends as the corpus luteum stops producing its hormones known as progesterone. The term menarche refers to the first uterine cycle, which begins at puberty. Again, average age of onset is between 11 and 12, but again can vary from female to female. Menopause refers to the termination of the uterine cycle, and average age of menopause is between 45 and 55. Amenorrhea means the absence of the cycle. And there are two different forms. Primary amenorrhea is a failure to initiate menses. In other words, it never starts, or here the girl is 17 or 18 and has not had her first period. Transient secondary amenorrhea is an interruption of 6 months or more of the regular cycle. This is often caused by physical or emotional stress. Many female athletes, especially gymnasts, will undergo transient secondary amenorrhea. Next structure is the vagina. This is an elastic muscular tube that runs between the cervix and the vestibule. It averages between 7.5 and 9 centimeters in length. Remember that the cervix projects into the beginning of the vaginal canal. This creates an area known as the fornix, and this is up in this area here. The fornix is the shallow recess that surrounds the cervical protrusion. This fornix actually allows the gynecologist to be able to palpate the female reproductive structures and various organs and to make sure that everything is positioned, etc., for a woman to be able to deliver naturally. Functions of the vagina. This is the passageway for the elimination of the menstrual fluids. It also receives the spermatozoa during sexual intercourse, and it forms the inferior portion of the birth canal. Looking at the structures, the vaginal wall contains a network of blood vessels and layers of smooth muscles. There are no glands in the vagina. It is moistened by secretions that are coming from glands in the cervix, but there are no glands per se in the vagina. There is a hymen. This is an elastic epithelial fold that partially blocks the entrance to the vagina. This structure is typically ruptured by first sexual intercourse or often through the use of tampons. In the walls of the vagina, you will find vestibular bulbs. These are masses of erectile tissue that lie beneath the muscles. Here's one of those structures I talked about in the beginning. The vestibular bulbs have the same embryological origins as the corpus spongiosum of the penis. They are erectile tissue. Histology of the vagina. The vagina epithelium also will change with the ovarian cycle, not to the same extent as the uterus, but it will undergo some changes. The vaginal lamina propria is very thick and elastic, and this is where you find blood vessels, nerves, and lymph nodes. The vaginal mucosa consists of layers of smooth muscle fibers that are continuous with the uterine myometrium. The vagina also has a unique set of bacteria, part of our normal flora. This is a population of harmless resident bacteria that lives in the vagina. These bacteria are supported and survive through nutrients coming from the cervical mucus. These bacteria create an acidic environment that will restrict and prevent the growth of many other pathogenic organisms. They also, because of the acidic environment, this creates a very harsh environment and not very hospitable to the sperm. And this is why when we talked about semen production, that a lot of buffers and alkaline products are secreted into it or are part of the semen to help neutralize this acidity. Women will often have a vaginal smear done. This is where they take a sample of the epithelial cells that are shed at the surface of the vagina. These can be used to estimate the stage in the ovarian and uterine cycles. Again, trying to see if a woman's cycle is, especially if there's problems, say, with fertility or problems with your cycle, they can monitor, are you progressing through your cycle the way you should be? Next, we need to look at the external genitalia. This is known as the vulva or the penundum. This term refers to the area that contains the female external genitalia. Part of this is the vestibule. This is the center space that is bounded by small folds called the labia minora. This area is covered with smooth, hairless skin. The urethra opens into the vestibule, just anterior to the vaginal entrance. Also in the vestibule, you will find the clitoris. This is a very small protuberance, and this is another structure. It has the same embryonic structures as the penis in the male. There are three columns of erectile tissue in the clitoris, the same as you see in the male penis. And also extensions of the labia minora will form the prepuce, or the hood over the clitoris. There are also vestibular glands here that secrete into the vestibule near the vaginal entrance, providing lubrication. The mons pubis and the labia majora, these form the outer limits of the vulva. They protect and cover the inner structures, and they are primarily adipose tissue. You will also find sebaceous glands and apocrine sweat glands here that secrete onto the inner surface of the labia majora. Next are the mammary glands. They secrete milk to nourish an infant, and this process is known as lactation. They are specialized organs of the integumentary system, and they too are controlled by hormones of the reproductive system as well as the placenta. They lie in pectoral fat pads deep to the skin of the chest. We will look in our last chapter, we will talk more about how the mammary glands produce the milk and how the process of lactation itself. The parts of the breast, each breast has a nipple. This is made from ducts from the various mammary glands that carry the milk to the surface. The areola is the reddish-brown skin around each of the nipples. So here we can see the inside of the breast is divided into lobes. Each contains several lobules, these small structures, and the lobes are separated by this dense band of connective tissue. And you can see how coming out of each we have a duct, and all these ducts combine to form the nipple. The mammary gland ducts are what leave the lobules. They converge to form a single lactiferous duct in each lobe. These lactiferous ducts enlarge near their end and form an expanded chamber. Anywhere from 15 to 20 lactiferous sinuses open into each nipple. Now we're going to look at the hormones of the female cycle. In general, this involves secretions of both the pituitary gland as well as the gonads. And this forms a complex pattern that will coordinate both the ovarian cycle and the uterine cycle. This picture here, before we go through everything, in your textbook, there is one large chart that shows all the various components of what happens at various levels through the monthly cycle. I highly recommend that you refer to that whole picture in your book as you are going through these slides and as you are studying. I have kind of broken it apart into individual sections, but this makes a lot more sense as you are studying and going through it if you have the whole picture in front of you. Everything with the cycle begins with the gonadotropin releasing hormone that is released from the hypothalamus. This is what regulates reproductive function. The gonadotropin releasing hormone is released in pulses. So you see anywhere from 20 to 50 pulses per day. This is how this is released. As you can see from this graph, pulse frequency and amplitude change over the course of the ovarian cycle. This is controlled by estrogens and progestins. Estrogen will increase the frequency of these pulses, whereas progesterone or progestins decrease it. So again, if you have the whole chart in front of you, you would see that estrogen is prominent during the follicular phase, or the first half of the cycle. Progesterone is the prominent hormone in the second half. So we see estrogen, when it's prevalent, increases the pulse frequency. Progesterone will decrease it. Now we're going to look at how hormones affect the follicle. So hormones and the follicular phase. This begins with FSH stimulation. Remember, FSH stands for follicle stimulating hormone. This is going to stimulate the development of the follicle. Every month, some primordial follicles will be stimulated to develop into primary follicles. And we said before, every month, roughly, give or take, around 10 primordial follicles begin this process, but usually only one makes it to the point of ovulation. Now, the main hormone during the follicular phase, the first half of the cycle, the main hormone is estradiol. This is the most abundant hormone prior to ovulation. So again, we see here FSH will cause the development of the follicles, and this is where estrogen comes from. There are 5 main functions of estrogen. One is to stimulate bone and muscle growth. Estrogen also maintains the female's secondary sex characteristics. This includes body hair distribution and adipose deposits that create the typical female physique. Estrogen affects central nervous system activity, especially in the hypothalamus, where estrogens will increase the sexual drive. Estrogen helps to maintain the functional accessory reproductive glands and organs, and it initiates the repair and growth of the endometrium. All right, so again, in the follicular phase, here, the developing follicle. Early in the follicular phase, estrogen levels initially would be very low, and the gonadotropin pulse frequency would be somewhere between 16 and 24 a day. But as we approach the tertiary follicle, so as the tertiary follicle forms, the concentration of estrogen rises steeply, and the gonadotropin pulses will also increase to around 36 per day. Then we get this sudden surge in luteinizing hormone. This ends up triggering the rupturing of the follicular wall and hence ovulation. Ovulation occurs somewhere between 34 and 38 hours after the peak of luteinizing hormone, or after luteinizing hormone secretion begins, or roughly 9 hours after it has peaked. Once ovulation occurs, then we enter into the luteal phase. The luteal because the corpus luteum is in control. These high luteinizing hormone levels, as we said, promote or trigger ovulation. This will then promote the production and secretion of progesterone and trigger the formation of the corpus luteum. Now, because progesterone levels are high, the frequency of gonadotropin releasing pulses are going to start to be low. It's those higher frequencies of these pulses that is responsible for that surge here in luteinizing hormone. But once progesterone is in play, these pulses drop very low, and because there aren't a lot of pulses, there will not be a lot of FSH produced. If you think about it, we don't want any FSH right now. We just developed a follicle. It was just released. We are anticipating pregnancy happening. We don't need to develop another follicle. So the pulses will drop, and which means that the level of FSH. Look at the amount of FSH, this blue line here, compared to the follicle phase. Okay, very, very low. So during this luteal phase, progesterone levels will remain high for roughly 1 week. Unless pregnancy occurs, then after about a week to 12 days, the corpus luteum will begin to degenerate. And we'll, again, in our last chapter, we will talk about some of the, how does the ovary know? How does the corpus luteum know whether or not pregnancy has happened? We will get into all of this later. Near the end of the luteal cycle, progesterone and estrogen levels will drop off dramatically. This will now allow the gonadotropin-releasing hormone pulse frequency to increase. As those pulses increase, that is going to stimulate an increase, as we see right here, in FSH secretion. That means, okay, increase FSH, we're going to start another cycle. Okay, pregnancy did not occur, so we're going to start another follicle developing. All right, here's where we see the estrogen and progesterone levels. Estrogen being very high during the follicular phase, and progesterone being very high during the luteal phase. Now, how do these hormones affect the uterine cycle and what's happening at the level of the uterus? When the corpus luteum degenerates, progesterone and estrogen levels decline. And this is what results in menses, results in the deterioration, destruction of the lining that we talked about earlier. So the endometrial tissue will begin to shed, and it will shed for several days. And it continues, we see it shedding here, and it will continue to shed until we start to see a rise in estrogen levels. These rising estrogen levels will stimulate the regeneration of the functional zone, and this is known as the proliferative phase. The proliferative phase continues, okay, until progesterone levels begin to rise. And as progesterone levels rise, this is when you enter into the secretory phase. This increase in estrogen and progesterone will cause enlargement of the endometrial glands and an increase in secretory activities. Hormones also have an effect on body temperature. Monthly hormonal fluctuations will affect our core body temperature. And some women actually use this as a means of birth control or timing when they can predict when they would be the most fertile in their cycle. During the luteal phase, which is the second half over here, remember progesterone is the dominant hormone. And look at the body temperature, slightly higher during this phase. And during the follicular phase, this is when estrogen is the dominant hormone, and the basal body temperature averages about 0.3 degrees centigrade lower than during the luteal phase. When ovulation occurs, there is this initial drop in basal body temperature, and then this abrupt spike or rise in temperature after ovulation. And so if a woman is trying to predict and figure out when ovulation occurs, what you do is you take your temperature first thing in the morning before you get out of bed, and you monitor the temperature. The day your temperature goes up, okay, you know you ovulated within the last 24 hours, and this would be the time when you are most likely to get pregnant. You can also figure out when your period is going to start. You wake up in the morning, taking your temperature, and it has dropped. You can be sure that if it hasn't started already, it will start before the end of the day. Ending with some sexually transmitted diseases. These are diseases that are transferred via sexual intercourse. They include bacterial, viral, as well as fungal infections. One is pelvic inflammatory disease. This is due to the bacteria known as chlamydia. This is very serious and can result in infertility. AIDS is due to a virus. Gonorrhea and syphilis are both due to bacteria. And the last three, herpes, genital warts, and cankeroids, are all viral infections.